Oh no, can you hear me now?
Yes, doctor. Yes, doctor.
Okay, cool. How you can hear when the humming happened to me? Okay, Yeah, I don't want to listen too much because I hear my voice sometimes I cannot talk. Okay. Sorry about that. I try to repeat to you all again. Let's say I turn off this should be okay, right? Hello, can you hear me?
Yes,
Steven, can
doctor.
you hear me? Ah, good
Yes,
idea. Yeah,
doctor.
because I'm going to turn off the Bluetooth. Okay, now I try to repeat again because this I'm so sorry. It was a bit lagging, right? Okay, the primer design, which means that you need to focus on the length of the primer. You need to focus on the melting temperature. Melting temperature, which means that when the DNA itself, they are split apart like this, we ensure that they are not going to split for the whole thing. So this is let's say this is the length of your genes. So the TM temperature at 55 degrees, they will they will try to tear off the DNA partially. All right, they're going to tear off entirely. So this one is what we know as a TM temperature. Basic concept lah, huh? Okay. So also we need to focus on the GC content. You may see that the whole DNA, there are combination of 4 different nucleotides only. They are either to be A, T, G, or C. But if let's say, as we know that for the A and T, their binding strength is much lesser compared to G bound to C. Why? Because T and C, they are duplex. G and C, they are triplex. So that's why there are 3 different. You don't need to put this in your thesis, but I explained to you today before when you accept that. So because GC is much strong, the binding of the GC is much stronger compared to the AT. That's why we need to make sure that the GC content cannot be too much. Usually in the primer design, we can only set it up to like 60 to 65 degrees. Too much of the GC contents, it will lead to this kind of the scenario because as a hairpin. All right, hairpins, which means that the DNA of that particular gene, maybe one of the end, they bound on back onto their own gene again. They bound onto their DNA. That's why this DNA, you cannot run the PCR anymore. So because you can see, usually it need to be open-ended. Once turn out to be like something like this, they bound onto the back. So this, when the primer run to the certain segment, then you stop. All right, so this is a case that we need to be aware of. Okay, now, after using the primer design, just now maybe you missed out something, but don't worry. So I try to use the whole thing of these nucleotides from the Giardin one. I put into the primer 3 just now. I just use their own default system. I set it to 200 nucleotides long, the length. So of course now they can give me something. The length itself is like they're covering from the 159 to 368. All right. So this is quite okay. So this is the segment is covered for you. Okay, all right. So this is quite good. And then you may see the pair of the primers sent over here. So you have the CAT, whatever. So it copy, oh no, sorry, it copy from this segment until to this segment. So this is a reverse complement for all the primers. So this is the another primers. So you may see because after reverse complement, the whole segment of here, they looks totally from this. Say for example, you can look like from this TG from the back, from the end there, T, G, C, and T. But here you may see that T is complement to T, C is complement to G, and so forth and so on. Okay, just like that because, and then it has a pair of primers. So your things, it can, when you do a PCR, it can cover the whole thing. Okay, now, at least now you can understand this. I want you all, those who are working on the Bamani and the Shigella, especially these 2 groups, and I will send you a list of the protein, of the genes. What you need to do your first tugas and your assignment today is you get the gene. I'll show you your accession number. Then you go to the NCBI. You're not going to do the primary. You can try. Don't worry, you can try. But you just go to the NCBI. Then you try. Now I will share with you. I put onto the list here. I will show you one of the paper here. 1 second, I keep that. Okay, these are a couple of paper that I found. This is quite a good paper. Okay, this is a paper pretty similar to like what I proposed you to work on that, but we work in a different form. This one is published in Brazil. You can see about money happen everywhere, you know. You work about money, huh? One day the WHO will come to see you. Because about money is something very challenging. It's like a TB. It doesn't have any drug anymore. You know that just three isolate that you're working on, one need to, out of the 10, 3 of them that's resistant to all the drugs in the market. Just imagine if one patient and admitted to hospital, check with the bar money, and all the drugs on the pharmacy, the rack, they are not working. It's quite risky, you know. So that's why we have to be aware of this. Okay, so this is something like I will put them in a good form for you. You can see like from this paper, it's saying that they're using the human serum infected with the Bamani. They use a human serum and then they try to do something we call as a proteomic study. They try to, you know, they use this form. Yeah, more complicated, something like that. Okay, maybe you should look at this or how can so many dots on you don't know why it's like, okay, don't worry about that. This is to show that each dot of these, they are the proteins. They are each, what does it mean by the protein? They are the reactive proteins against the human serum. As you know that in our concept, when someone infected with that particular, say for example, Bamani, come to the body, then our immune cell recognize them, they will start to attack them. At the same time, I think you all already studied about this basic immunology, right? Our B cell will recognize them and B cell will release the antibody. And also at the same time, they will generate the memory cells. Correct or not? And that's why this serum already contain those antibody released by this patient serum, this in the patient infected serum. So that's the serum itself can recognize what does it mean with the Bamani anymore. So they know. That's why you need the vaccine. Understand or not? Before someone getting the vaccines, our body doesn't have any particular T cell or B cell can recognize the particular pathogens. But once someone infected with that disease, body will raise the antibody, whatever. That's why we need the vaccine for prevention. Sometimes the disease itself, for example, like certain things that are quite acute, then people can, like for example, COVID-19 is one of the example. The virus itself, they will not kill the human, but it's caused by our own, we call it as a over response of our immune system, or because over inflammations, the cytokine syndrome, acute cytokine syndrome, they kill our own human. So in this case, luckily Bamani, it will not cause that, but it still will cause the immunocompromised patients into a problem. All right. So that's why this every single dot of here is supposed to be the reactive proteins against the infected serum. If you compare to the normal serum, maybe the normal serum, those never infected with the seed in the Bamani one, it will only show the clear things only because it doesn't have antibody against the Bamani. So this one, because when you run this, if it means that you run the Bamani onto the SDS phase first, onto the protein gel, then you do a Western blot using the serum to blot against the Western blot. So you will see that, okay, these are the things. Okay, now I don't want to tell too much of story, so we understand that very soon. Don't worry. When it comes to the lab, then we understand. I worry about the students, especially those online, because they're not here. I cannot show something using my hand. Okay, I hope that you can see from me. Can you see me or not? Hey, where's the camera? Over there. Okay. Can you see me or not? Oh, because I'm studying, you cannot see me, right? I'm sorry, you cannot see me, Okay, don't worry, But okay, for those working on the Bamani, I will try to highlight these couple of things for you. For is something your homework because I cannot tell you everything. If I share with you everything, everyone will somewhat get yawning. Very boring. Okay, I let you to think, then you understand. So their OMPA ultimate marine A, OMP 34K. I think this is should be the Ton B lah, similar to Ton B. OPRC, there are a couple of these. So what you need to do is, I will give you some homework today. You can, those working with my money, sit together, get their nucleotides and get their protein out. Do something, and then try to do for your what? Primer design. I will still to do that, don't worry. I will cross check. But I believe in you. I don't say that I don't believe, but I want you to try to do that. Okay. For the one who working on the Shigella, we publish Shigella quite a number of paper already. So I will also share with you what kind of Shigella we need to focus on. 1 second. Completion. Okay, this is one of the paper published years ago by a master student of Cairo. So you may see that there are a couple of different iron binding proteins. Those who working with the Shigella, I think they are divided into two groups. One group is working on the outer membrane, then the other group is working on the iron binding proteins. Okay, so I will share, similarly, I will share something to you all also. So what you need to do is you need to go and try to get out where can you find this the gene and the nucleotides. Just like that enough. Then we have a chat again. Hopefully once you get it done, then you WhatsApp me, then you can send to me. So I will go through. Understand or not? Okay, another part, those who are working for the mushroom part, okay. I will share with you because for the mushroom one, actually ours one is quite established already. Because we need, don't say we working on mushroom. I don't know how to grow it also. But Dr. Heng knows it very well. He knows it very well. I think that one can make money one. So this one, but because my part is only to study about the immunological response. So he and the counterparts will provide us the polysaccharides. So like you all, like those working with the polysaccharide one, you start to with the polysaccharides. Then we will try to treat it onto the THP1 and see what's the response. Actually, this is a part of the industrial collaboration project. All right, what you do now, we're going to share the data together with the industry later. Okay, so now I have something to share is we have a paper published this before. I'll share this paper to you. I'm not going to throw everything to you, but I will to share with you that what are we going to do with that? Okay, 1 second. Put by size. Okay, this is a paper just recently accepted. Okay, you see what we are doing over here is we already have all the primers designed. Basically, we focus on this couple of different genes. But in this case, maybe I want the current student to work on one more genes, which is IL-10. This is very, very important genes. This like interleukin-1 beta, interleukin-6, these are all the cytokine. All right, I think Jia also working on this cytokine, right? And then I have all these things already because someone is working on the human. We have those things, but if you want to learn, you still can try to go into the database, get the genes out and see how it is designed. However, when we design this, we also try to compare it with the validated data. Actually, primer design is not your concern part. Your concern part is an outcome only. You're a pharmacist. You're not a scientist. I'm going to train you. If I want to train you, you want to learn more, I'm most welcome. You can come join me for your master PhD. But because as FIP, we try to focus onto the important thing first. All right, we don't have too much time to go into every single things. And some of these things are very well established one. We not necessary to go into too detail. Okay, and then of course, one more thing is we need to have one is a housekeeping gene, which means that what does it mean with a housekeeping gene? Like for example, we can use a GAPHD, or sometimes people will use a tubulin, sometimes people will use actin. Housekeeping gene, which means that something it is expressed evenly. No matter you eat fat, you eat sugar, in what conditions, they still appear evenly, the expression. That's why we call it a housekeeping gene. It needs to be there stably all the time. It cannot up and down. It's up and down, we cannot claim it as a housekeeping gene. Then you ask me, how can we know it's housekeeping gene or not? Don't ask me. This has been selected. So if let's say you manage to go into someone body again, can you have your cheek cells, give me some hairs, run some genomic, then you have to sit there and run this is the housekeeping gene. Housekeeping gene you have to do a lot of validations, give them a lot of treatment to see whether the expression must be there all the time. If let's say they never change to a certain extent, that's why we call it housekeeping gene. Why do we need to have housekeeping gene in the system? Because everything the same. Today, you want to show that how young you are. You need to have a control. Correct or not? You can use me as a control. So you are very young. No, you are much older, maybe. Just like that, you understand or not? You want to compare with something, you need to have a control to compare with. You cannot simply say, I'm younger, like in the test online. You eat this, you can see a lot of things. I hope that in the future, you are a pharmacist, you can try to do more education for the society or community. Because I found that now on social media, there are lots of the misinformation. You eat this, then you become better. Even though I saw a company, you plug your hair and then go to run the DNA, then they will know that whether you belongs to the smart one, you belong to a stupid one, belongs to a handsome one, belongs to whatever one. And come on, DNA cannot tell you everything. They can tell you about your genetic background only. But in DNA cannot tell whether someone has a potential to be cancerous or not. No, never. That's why these are all the misinformation run by the commercial company. But sometimes I want to raise, we cannot raise. I don't have money, they have money. They sue me and you're trouble. USM will be trouble. That's why I understand or not. But as a pharmacist, that's why you come to learn all this. This is a future trend, Pharmacists, you need to learn all these pharmaceutics, all these things. But you can see when AI coming in, artificial intelligence coming in, when you come to the drug design, everything think about is a gene only, because eventually the drug must come into the human body or the vet or the animal body, because either the vet or human only. Understand or not? So that's why now these four biological data and AI, they are so close to each other. And somehow, if you don't believe it, even yesterday I tried to use ChatGPT. They can help us to do the primary design also. AI, because as long as something with the alphabet, as long as something with the data is there, ChatGPT or DeepSeek is quite useful. So you can try to use, which means that it can help you to try to do a sort this thing. Okay, now, why do we need to have a housekeeping? Which means you need to compare. If let's say you load everything in, but firstly, you do not know how much that you load in, it turn out to be your data could be like valuable. So housekeeping gene is something that you need it as a compare. When you do the normalization, you will normalize it against housekeeping. Don't worry, this one I teach you when you come to data analysis. At least I show you what does it mean over here. Okay, you look at our students. So she managed to finish up all this thing is, I tell you, if my student can finish this, you all can finish this also. Because now Munira, she's a student of mine. She's working with one of the company. Is a global clinical research company, quite established one. I hope that in the future you all can try to think about this also. Not only a KKM itself, a lot of opportunity out there. Okay, so you can see that, for example, this one I will show you a paper later lah. So when you get the data, then you will know that you will have this kind of like a scenario. Okay, this is after analyze, though this one we call as a gene expression. Then we're going to compare with one group of the cells treated only with the PPS. One group of the cell may be treated with the LPS only. LPS, why do we need to use the bacteria LPS? Because we already has been known that LPS will trigger cell to lead to the inflammation. So LPS we usually use as a positive control. Then you want to compare your things whether they're working like LPS or not. Of course, then you have to do the treatment. That's why you can see this is different concentration, and the paper showing that we have 24 hours, and the other one is 48 hours. Understand or not? So for everyone, the students, so those working with the treatment, because at the polysaccharide response, so it's not complicated. So usually we set it at we start with a 0, followed by 6 or 4 hours. We will plan that later, and then 24 hours and 48 hours. Then close everything. Then you can start to do your analysis, similar to the infection. See, similar to all Ivan, same also. Ivan here and Ziying almost similar, right? Or Ziying and Jia Yi, huh? Yeah, Nelson, right? I need to, after submission, all the mark, all I seems I'm on all my brain also submitted to the whole office already. Okay, just like that. Don't worry. Same also. Now, can you understand about what I want to mean or not? Okay, now we round table question. I don't want to give you too many things. Otherwise, 2 o'clock now, you'll be confusing. We round table, any question you ask me now. Then I will, anything else, then we'll come to the next week we come for. We need to have more regular meeting from now. Okay or not? All right. Okay, I've stopped sharing. Then students, we can have some chat over now. I will share all these things, the paper information to the group later. Yes, student? Any questions? Don't be shy. It's come to your fourth year already. Can you hear me or not? Chunket, any question? Yes, Chunket.
Dr. So far, no.
No question, right? San Leong?
I also no questions.
Safra?
No question, doctor. Thank you.
Chen Ying? Chen Ying? Nelson?
Doctor, for my part, right, about money, right? Yes.
Yep.
Can you hear me, doctor?
Yeah, I can hear you. I can hear you. Don't worry. Please carry on.
Okay, doctor.
Yes.
Doctor, for my part, right?
Mm.
The about money, right? We need to focus on which one, the outer membrane OA or outer membrane 34 kDa. Which
Okay,
which
because
one we
there are a
should
couple of
be
different
like focus
money.
in?
Yours one, I think, because you are not focused on recombinant protein. You only need to start to study about the genes expression from the Barmani, right? Correct or not, Chen Ying?
Yes.
Yeah, we're going to
Doctor.
focus on a couple of them, maybe 3 to 4, not only two.
Yes.
It's not enough. We need to focus on a couple of them. I think there are up to 6 only. Maybe we can try to study them, because once you run the daytime PCR, you can finish up the 16 together in one round. Okay or not, Chen Ying?
Okay, thank you, doctor.
Don't worry, I will still have a discussion next week. We still can have a, but you need to do some homework only to you all. Once I give you all, so you stick back to your own your title. Those working with the TSP1 with the polysaccharide, don't worry, but you can train yourself also. All right. So what about now we come to the next one. Nelson, are you okay? Good. Zhou Ying?
No question, doctor.
Very good. Jia Yi is here. Jia Yi, no question. Later. Xiao Rou is here. Okay, Yu Rou is, I see. Okay, Ziying is here also. Winnie is here also, right? Yeah. Who else? Just now. It's me. Last one is me. Okay. I think I asked. Oh, Vin Yuan.
Yes, doctor. So far, so good. Thank you, doctor.
Wan Ling.
Yes, doctor. I'm good.
Very good, right? Don't worry. Hey, Eric. Yeah, Eric. Where's Eric?
I'm here, doctor.
Oh, Chen Zhu is here, right? Oh, Chen Zhu is not here also. Sorry, Chen Zhu is Chen Zhu, did I ask you just now? Eric, okay, huh? Okay, so far,
Yes, good,
Eric,
doctor.
thank you so much. Now I start to not just give me another 5 minutes. So what you need to do now is for FIP, don't rush, do it step by step. You don't understand, you must ask. Oh, and then I know there's still a lot understand. If I were in your situation, I really can understand. All right. But that's why I assign a mentor to you all. I know my mentor Jakala, he's going back to his position. He'll still be back next week. And then I asked them never design some complicated experiment for you all. First thing is we need to ensure the time is within. is there. Another thing is try to take care of my money. My grant is so limited now. I cannot afford to do so many complicated things. But I'm happy. I want to ensure that you all come up with a very good thesis. All right. Not only like that, I want to have a publication from each of you. Some of them maybe I will combine. I want to ensure when you get graduated, you can have your name to show to your employer you have something like that. This is very useful for you now. Everyone can get a degree. You come to BFARM, you will get a first class degree. You go to maybe UM, but I want you to make something unique from other. Understand? I cannot promise saying that you will get your publication before your graduation. At least we have something to come out first. Then when you come to your work, then I will try to push out within a year. Like for example, my student Munira, they all got their publication. She's so happy that she got the paper. She shared it to her employer. I said, but it's good thing lah. So happy ma. It's a Q1 paper. It's very nice paper, you know. But we try a lot of put in a lot of efforts. I believe she can do that. You all definitely can do that. Understand or not? Oh, success like that. It's about the efforts only. So keep this good effort in your future career. Okay, the online one. All right, I'm going to stop here for the online. Then thank you so much. Then I will we catch up hopefully by next week again. I will share something on the group, so hopefully you can try to complete that the works. I will share the paper to you all. I don't want to say last time because I cannot explain. Thank you so much. Bye bye.